Welcome to Biology at Priyajit, my dear viewers. In this class, today we will explore eukaryotic transcription step by step, focusing on its three main stages: initiation, elongation, and termination. Transcription is basically the process by which genetic information in DNA is copied into RNA with the help of specialized RNA polymerase and transcription factors. By understanding these stages in a simple and systematic way, we will see how eukaryotic cells precisely regulate gene expression for efficient transcription. Transcription is basically the process of RNA synthesis from a double-stranded DNA template. During this process, the two DNA strands separate from each other by breaking the hydrogen bonds, and one strand act as the template. The enzyme responsible for DNA dependent RNA polymerase because it synthesizes RNA using DNA as a template. Stages of transcription. Transcription in eukaryotes occurs in three stages: initiation, elongation, and termination. This is the three stages of eukaryotic transcription. Some differences between prokaryotic and eukaryotic RNA polymerase. We have already studied about the prokaryotic RNA polymerases. Now we will know the characteristics of eukaryotic RNA polymerases. In case of prokaryotes, the RNA polymerase consists of 4 subunits and a sigma factor. But in case of eukaryotic RNA polymerase, there are 5 different types of RNA polymerase. In case of prokaryotes, only a single type of RNA polymerase is needed. In eukaryotes, there are 5 different types of RNA polymerase. The core enzyme in case of prokaryotes contain 5 subunits. Every types contain 10 to 12 subunits in case of eukaryotes. In case of prokaryotes, the molecular weight is around 400 kilodalton, but in case of eukaryotes, the molecular weight of RNA polymerase is about 500 kilodalton. Types of RNA polymerase in eukaryotes. RNA polymerase 1 transcribes 28S rRNA, 18S rRNA, and 5.8S rRNA. RNA polymerase 2 transcribes mRNA, small nuclear RNA, and microRNA genes. rRNA 3 polymerase 3 transcribes tRNA, 5S rRNA, small nuclear RNA, small cytoplasmic RNA, and U6, that's a special kind of small nuclear RNA. So this is the table, and there are different types of RNA polymerases. Additional RNA polymerases in plants, there are RNA polymerase 4 which synthesizes some silencer RNA, and RNA polymerase 5 is involved in heterochromatin formation. So there are different molecular components of eukaryotic RNA polymerases. This is the table showing the different subunits like beta and beta dash like subunit, alpha like subunit, omega like subunit. There are 4 common subunits in 3 RNA polymerases, and there are enzyme specific subunits, and there are total subunits. There are 14 subunits total in RNA polymerase 1, and there are 12 subunits in RNA polymerase 2, and there are 16 subunits in RNA polymerase 3. So what are the transcription factors? RNA polymerase functions with transcription factors. For RNA polymerase 3 and 1 and 3, few and simple factors are needed. For RNA polymerase 2, many factors collectively called as basal factors they are needed. Basal factors plus RNA pol 2 together they called as basal transcription apparatus. Organelle RNA polymerase, mitochondria and chloroplast RNA polymerases resemble bacterial RNA polymerase. So their genomes are small, so transcription regulation is very simple there. Effect of alpha amanitin. RNA polymerase 2 is highly sensitive and it is inhibited at low concentration. RNA polymerase 1 is not inhibited, and RNA polymerase 3 it is inhibited at high concentration in animals but not inhibited in yeast and insects. RNA polymerase 1 and CTD tail of RNA polymerase 2, there are two largest subunits, RPB1 and RPB2, and RPB1 contains C-terminal domain or CTD tail, and this CTD tail consists of the repeats of the heptapeptide tyrosine, serine, proline, threonine, serine, proline, and serine. Number of repeats in yeast, the number of repeats is 26 to 27. In case of mammals, there are 52 repeats, and in case of eukaryotes, there are 26 to 52 repeats. So functions of CTD, they are essential for cell survival. There are minimal 10 repeats needed in yeast. These sequences are unphosphorylated during initiation, but the tyrosine and serine residues become phosphorylated during elongation. It regulates transcription and RNA processing. Now we should know about the elongation process of transcription, about 30 nucleotide synthesis. After about 30 nucleotide of RNA are synthesized, RNA polymerase leaves the promoter and enters the elongation phase. Most transcription factors remain at the promoter sequence and can rapidly reinitiate the transcription with another RNA polymerase. So the molecular structure and functioning of eukaryotic RNA polymerase 2 during elongation were revealed by Roger Kornberg and colleagues, for which Kornberg received the Nobel Prize in Chemistry in 2006. During elongation process, RNA polymerase maintains a transcription bubble in which 8 nucleotides of RNA remain base paired with the DNA template strand. The DNA double helix now then enters a cleft of the polymerase and is held by jaw-like extension of the enzyme. So the two DNA strands are unwound and complementary ribonucleotides are added to the 3' end of the growing RNA chain. The DNA-RNA hybrid then bends sharply inside the enzyme, positioning the 3' end of RNA at the active site for further nucleotide addition. The newly synthesized RNA is then separated from the DNA template and it exits through another channel of the RNA polymerase. Termination of transcription in eukaryotes. What happens in case of RNA polymerase 1? It requires a specific termination factor. The factor binds to the DNA sequence and downstream of the terminator site. So functionally it is similar to the bacterial Rho factor but binds DNA instead of RNA. So this is the transcription process, transcription termination by polymerase 3 and Pol1, which uses specific terminators to end the transcription. But in case of polymerase 2, the 3' end formation of Pol2 transcripts facilitates the transcriptional termination. So we know what happens in case of RNA polymerase 2. RNA polymerase 2 terminates after transcribing a poly-U sequence in RNA, which is similar to Rho independent termination of bacteria, but it does not require that kind of hairpin structure before the U-rich region. RNA polymerase 2. This is the RNA polymerase 3, and what does RNA polymerase 2? This is torpedo model. So termination does not occur at a fixed DNA sequence. RNA pol 2 continues transcription hundreds or thousands of nucleotides beyond the coding region. The pre-mRNA then is clipped at a specific consensus polyadenylation site while transcription is still ongoing. Then this cleavage produces the mRNA for protein synthesis, a downstream RNA fragment whose 5' end remains attached to the polymerase. A 5' to 3' exonuclease called RAT1 in yeast, which binds to this free 5' end. Then RAT1 degrades the RNA while moving toward RNA polymerase like a molecular torpedo. When RAT1 catches the polymerase, the transcription complex dissociates and termination occurs. So as we conclude, eukaryotic transcription is finely regulated process involving specialized RNA polymerase and multiple transcription factors. From precise promoter recognition during initiation, controlled RNA chain growth in elongation, to accurate RNA release during termination, and each step ensure faithful gene expression. Mastering these stages is essential for undergraduate, postgraduate and NEET preparation and for understanding how genetic information is converted into functional RNA in eukaryotic cells. So keep learning and exploring the elegance of molecular biology. Thank you.